This tutorial is for the basic information module of the Integrated Microbial Genomes Annotation Collaboration Toolkit. The first thing I need to do is log in, and I do so as a student, into Genetics Biology 262. In my course, Biology 262, I have an assignment that has been given to a student team. One of the quirks that is part of the Annotation Collaboration Toolkit is that every student is a member of a team, even though you might be the only member of your team. So you are a team of one. I'm going to click here on the assignment. Now you'll see that in this assignment I have been assigned 3 potential protein coding sequences that represent genes from a machine annotation or a machine examination of the genome of Thiomicrospira crunogena XCL-2. At this point, in order to complete all of the assignments in IMG-ACT, I must be comfortable with the notion of using tabs in Firefox. So the first thing that I'm going to do is point out that I will open this link in a new tab. You'll see that it pops up in the top window here, and we're seeing that I now have the laboratory notebook. So if we step back here to the assignment, we're going to compare this again to the notion of a regular laboratory where you make observations in your laboratory notebook about data that you observe in the world, and you finally come over here and make a lab report. So in this particular assignment, we are going to be collecting data and recording that data, basic information about a gene. And in this case, we're going to be filling in these boxes. So I'll take you back to the top, and I will point out that right here you can then see this information, and the same place is right here. So I'm going to open this in a new tab, and I'm going to now be connected to the Integrated Microbial Genomes /edu website. For those of you who were wondering why we didn't have lab last week, it was because this website did not work. So at this point in time, I will also draw your attention to the lab report link, and that one will pop up right here. Now it'd be nice if this said lab report and this said lab notebook and this said, my course, so that you could navigate without being caught up in some of the details, but that is the way it is at this point in time. So you can see that there are some fields that you'll be filling in for your modules, and those fields you'll be filling in from your laboratory notebook. So at this point, I'm going to move the tutorial on directly to the basic information module, and I'm going to, at this point, close my lab report so that I can focus in on what I have to do for my assignment for this course. So one additional item here is that you can see there's a window within a window in this Wikipedia wiki type format. We have some instructions, so you have almost like a miniature editable notebook inside here. So we're going to go back, and it says here that we need to get our DNA coordinates, and we need to enter those in our lab report. So I need to put some DNA coordinates right here. I'm going to get DNA coordinates from my gene details page. Whoop, and I'm going to get those DNA coordinates right here from this field called DNA coordinates. I'm just going to highlight those. I'm going to highlight the number, and go back to my lab report. I'm going to paste them right in there. So that sort of as a learning moment right here, you recognize that that's a very large number, 2,100,000 11,466. Yeah, so if you're wondering why and how that number got so big, we'll come back to that in a minute. Another key point is the minus sign, the recognition that double-stranded DNA has has 6 different reading frames, 3 in a positive direction and 3 in a negative direction. So we're going to simply, I'm going to make a note here because I know that's in my lab report, and it's not something we put in our lab notebook. So I can just type in here, this gene is noted to be in the minus strand reading frame. So that's just a note I made to myself. Now, to go on and further analyze whether or not this is a gene and how it works, we're going to need to gather some information. And one thing I can see right here is there's a hyperlink that says 1077 base pairs. I'm just going to click on that hyperlink and open it in a new tab where it says gene export. And here we're going to see that it has some really cool features. I'm just going to highlight it all, noting that I've taken all of the letters, the nucleic acids, as well as this caret, or this greater than symbol, and this information up above, and I've copied that. So another way to copy is Command C. I'm going to go back to my, DNA sequence where it says FASTA format, and I'm going to paste that right in there. So FASTA format is this piece of information. Whatever is on this line right here will be included, and down here now the sequence. I can remember, of course, that it said 1077, and so I'm going to just put 1077 and put that right there. At this point, I'm going to save my work because I'm a paranoid person who doesn't want to have to repeat what I've done over and over again. Now we're collecting this basic information. I've got this 1077, and you might make a note that you could take 1077 and divide it by 3, and if you divide it by 3 you get 359, yet there are 358 amino acids in this particular polypeptide. I'm going to open up this basic protein information in a new gene details page, and you're going to see a similar output, only you don't see A, T, C, and G, but you see a single letter amino acids. And again, I don't need you to understand precisely what that is. I'm going to close this one out, and I'm going to carefully highlight. The FASTA format for the amino acid, or polypeptide or protein sequence, pasted in there. And as I singled out, I recognize that this is 358 amino acids. I'm going to save that. Okay. Now I've completed the basic information module, and at this point in time, I can close off my gene details pages, go back over here, and recognize that I have a lab report to work on. And I can very quickly, because I've already been there, put in 358, and I can click the minus strand, and then also I can whiz back here, do some whiz bang, and I can copy this and go back over here. And I'll just take this. Now we're going to see some numerical problems. The smallest number, I made a little mistake there, just because it's the way we do our arithmetic, and I'll just cut that, put it right there, and I've got this one other piece of information called isoelectric point. I can say, oops, I didn't get that, so what do I do to get that? I can, in fact, use this link, or, which is the same as this link, or, which is the same as this link, and this link right here, to open up that gene details page and get that isoelectric point down here in the peptide. Right here I'm going to have peptide statistics, and so I can pop that page open. And the isoelectric point is something that the people who take biochemistry are very concerned about. It has a particular role with a protein. So we'll just pop that in there and say save. So I've saved my lab report for Module 1 Basic Information. I've finished off my Basic Information lab notebook. I don't need to keep the gene details page open. And so here I have completed basic information for gene annotation in this assignment number 1. And that means that that concludes basic information tutorial.